It is the most recognized villain in the history of human disease. Before viruses had names, before bacteria were visible under any lens, before anyone understood how sickness moved from one body to the next, there was the rat. Small, dark, fast, and everywhere. History handed it the blame for the single worst biological catastrophe our species has ever survived. 30 to 60% of Europe's entire population gone. Somewhere between 25 and 50 million people dead within 5 years. And at the center of that story, for 7 centuries, sat the rat. But the story history told was incomplete, and the part that was left out changes everything about how you understand what actually happened. To understand life for a rat in the 14th century, you have to start with the environment, because by almost any measure that matters to a small, opportunistic omnivore, medieval Europe was not a hostile world. It was an invitation. Medieval cities were, by modern standards, catastrophically mismanaged. There were no functioning sewage systems. Human and animal waste accumulated in the streets. Livestock moved through market squares daily. Food scraps decomposed in open alleys. And grain, the fundamental currency of medieval survival, was stored in enormous quantities in warehouses, cellars, barns, and merchant ships with virtually no pest control of any kind. If you were designing an ideal habitat for a creature that could squeeze through a gap the width of a coin, climb a vertical surface, eat almost anything, and reproduce every 3 to 4 weeks, you would design 14th century Paris. You would design 14th century London. You would design every major trading port from Genoa to Marseille to Bristol. The rat at the center of this story was almost certainly the black rat, Rattus rattus, also known as the ship rat. Smaller and more agile than the brown rat that dominates cities today, the black rat was an expert climber. It nested in the upper stories of timber-framed buildings, inside thatched roofs, within the wooden beams of merchant vessels. It was a social animal, living in dense colonies, raising litters in the dark spaces between walls that no medieval human ever inspected. A colony of black rats in a 14th century building was essentially invisible to the people living one floor below. They moved at night, fed in the dark, and retreated before dawn. Life in 1340, for a rat, was genuinely stable. Abundant food, reliable shelter, limited predation. The architecture itself was almost custom engineered for their benefit. Timber framing full of gaps, thatched insulation perfect for nesting, cellars connected to street-level drainage channels. Black rats crossed from rooftop to rooftop without ever descending to street level. Their entire world existed in the spaces between human structures, close enough to access human food, invisible enough to operate without interference. Ports were the highways of this civilization. Ships were essentially floating rat colonies, enclosed, warm, stocked with grain for long voyages, with mooring ropes that functioned as perfectly designed boarding ramps. The black rat's affinity for ships was not accidental, and it was ships that would carry everything that came next. The reproductive mathematics were staggering. A single female rat could produce a litter of up to 12 pups every 3 to 4 weeks. One colony could expand to fill an entire city block across a single warm season. By 1347, the rat populations of major Mediterranean port cities had been building for generations. They were, by any measure, enormous. And then, from somewhere along the Silk Road trade routes, from Central Asia, from the steppe regions connecting China to the Black Sea, infected cargo arrived. The mechanism that set everything in motion was not the rat. It was smaller. It was something the rat carried without knowing it carried anything at all. The oriental rat flea, Xenopsilla chiopus, was a permanent feature of rat life. Every rat in a medieval city carried fleas. This was simply the baseline condition of existence. Rats spent considerable time grooming themselves and each other. It did not work well enough to matter. When a flea fed on blood infected with Yersinia pestis, the bacterium that causes bubonic plague, something specific happened inside its digestive system. The bacteria multiplied. They formed a physical blockage inside the flea's gut. When the blocked flea attempted to feed again, it could not swallow. Instead, it regurgitated bacteria-laden blood directly back into the bite wound. The infection was delivered not by the bacteria moving forward through the flea, but backward, into the skin of the host. The rat had no immunity to Yersinia pestis. None. The bacteria entered the lymphatic system and began multiplying in the nearest lymph node. The node swelled, becoming a hard, painful mass beneath the skin called a bubo. Hemorrhaging beneath the skin produced dark discoloration across the body. Fever set in rapidly, and within 2 to 7 days of infection, the rat was dead. Archaeological evidence has documented mass rat die-offs that preceded the arrival of human plague cases by days or weeks. Medieval chroniclers recorded rats staggering out of walls and dying in open streets before human deaths began in the same neighborhoods. The observation was noted. The connection to what followed was not made. When infected rats died, the fleas living on them needed a new host. Fleas are not loyal. They respond to warmth and the chemical signals of a living body. When a rat colony collapsed, the fleas moved to other rats, to dogs, to cats, to the humans sleeping a few feet below on the other side of a wooden floor, sharing the upper stories of the same buildings with the infected colonies above them. The rat did not spread the plague through aggression, it spread it through death. The moment the rat died, the mechanism moved to the next available host. Now here is the part of this story that has been substantially rewritten by recent research, and that most popular accounts of the Black Death have not yet caught up to. The rat-flea-human chain is real. It is not in dispute. But researchers at the University of Oslo, publishing in the Proceedings of the National Academy of Sciences, constructed mathematical models of 9 separate plague outbreaks and compared how well different transmission mechanisms fit the actual recorded mortality patterns. Their conclusion was striking. In 7 out of 9 cases, the outbreak data fit a model of human-to-human transmission via human body lice and human fleas significantly better than it fit the rat-flea model. This matters because of scale and speed. The Black Death moved through European cities at a pace that is difficult to explain if you rely entirely on rats as the primary vector. Rats move, but they do not move as fast as trade networks, pilgrimage routes, and the daily movement of human populations. Human body lice live in clothing and bedding. In a 14th century urban environment where bathing was infrequent, clothing was rarely washed, and large numbers of people slept in close quarters, body lice were nearly universal. Research published in PLOS Biology in 2024 conducted at the NIH's Rocky Mountain Laboratories demonstrated for the first time that Yersinia pestis can infect the glands of human body lice and be transmitted through louse bites, a transmission route that had been theorized but never experimentally confirmed before that study. What this means is that the rat may have been the original reservoir. The rat-flea chain may have been where the plague entered human populations at the port. But once inside densely populated human environments, human parasites may have carried it the rest of the way. From body to body, household to household, city to city, at the speed of human movement rather than the speed of rat movement. The rat has spent 7 centuries as history's designated villain for a catastrophe it may have only partially caused. The daily existence of a rat in a plague-struck city was not a story of a creature aware of its role in anything. Black rats spent daylight hours in their nests, compact, warm, built from shredded cloth and straw, piled together in the dark spaces of walls and ceilings. Dominant males occupied the most protected positions. Subordinate males were pushed to the outer edges, forced to forage closer to human activity, more vulnerable to the traps and terriers and cats that medieval pest control could produce. At dusk, the foraging runs began. Grain stores were the highest value targets. But black rats were not selective. They consumed bread, dried meat, cheese, fish remains, candle wax, leather, and in times of scarcity, each other. They contaminated far more than they consumed, leaving behind waste that caused separate waves of illness, gastric disease, leptospirosis, and other rodent-borne infections running parallel to the plague itself. The predator pressure on rat populations was lower than it should have been, for a reason that intersects with another piece of medieval history. The Catholic Church, in the century preceding the Black Death, had formally associated black cats with witchcraft in several papal documents. This drove widespread persecution of cats. Historians debate how extensive this was, and the degree to which it affected rat populations remains contested. What is not contested is the basic ecological relationship. Reduced feline predation allowed rat populations to expand, and expanded rat populations meant a larger reservoir of flea-carrying hosts when the plague arrived. When the plague hit a rat colony, the collapse was rapid. Animals with no immune history died within days. the entire social structure disintegrated. Dominant animals died at the same rate as subordinate ones. Hierarchies stable for generations disappeared in days. Surviving animals dispersed, carrying their flea loads into adjacent territories, into new buildings, into new population centers. The plague moved through the rat population like a wave, and as it moved, it seeded the next generation of flea-to-human transmission events in each new location. Some rats survived. Genetic research has identified variants that conferred partial resistance to Yersinia pestis. Within a few generations of repeated exposure, surviving rat populations had a measurably higher frequency of resistance variants. This is natural selection operating in real time, within the same catastrophe that was killing human populations with no equivalent mechanism for rapid adaptation. Rats reproduce fast enough to replace their losses and accumulate resistance within years. Humans, reproducing on a timescale of decades, had no equivalent recovery mechanism. This is part of why rat populations survived the Black Death and rebuilt, and why human populations took more than a century to recover. As human populations collapsed, as entire villages emptied, as city districts fell silent, the rats' immediate environment shifted in ways that were simultaneously advantageous and catastrophic. Abandoned buildings became undisturbed nesting grounds. Food stores left behind by the dead represented unexpected abundance. Rats moved into emptied homes, unused granaries, deserted churches. In some locations, rat populations may have temporarily expanded on the resources left behind by mass human death, even as they were being devastated by the same pathogen. The rat did not understand any of this. It had no concept of the bacterium replicating inside its lymph nodes, no awareness of the flea feeding on its back, no knowledge that its death would send that flea onto a child sleeping 3 feet below. It was an animal, doing precisely what its biology had shaped it to do over millions of years, finding food, maintaining warmth, reproducing. The plague was a threat it could not perceive, could not respond to, and could not survive in the numbers it needed to. The Black Death reshaped European civilization across every dimension: economic structure, religious authority, artistic expression, the relationship between landowners and laborers, the fundamental assumptions people held about God and human suffering. It killed, by conservative estimates, 1 in 3 Europeans. It took parts of Europe more than 150 years to recover, and the animal at the center of the story, the creature that absorbed 7 centuries of blame, was a small, social, intelligent, adaptable omnivore that wanted nothing more than a grain store and a warm place to sleep. Recent science has not absolved the rat entirely. The rat-flea-human chain was real and almost certainly played a significant role in how the plague entered human populations. But the University of Oslo research, confirmed by the 2024 NIH laboratory work, has established something important. The rat may be at least partially history's most effective scapegoat. The plague spread faster than rats move. It reached places rats were not documented in large numbers. And the mortality patterns in multiple European outbreaks fit the human lice model better than they fit the rat model. The Black Death was not a punishment. It was not a conspiracy. It was a bacterium, moving through a world that had no knowledge of what bacteria were, no tools to detect them, and no means of interrupting their movement. It found the rat, it found the flea, it found human clothing and human bedding and the crowded sleeping arrangements of medieval cities, and it moved through all of them with complete indifference. Somewhere in the walls of a medieval city in 1348, a black rat with swollen lymph nodes stumbled out of its nest into the open air, lay down on the wooden floor of an upper story, and died, having never understood a single thing about its place in the worst catastrophe in recorded human history. The flea on its back had already moved on.